All right. Okay. 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 So generally in this class we'll be looking at the genetics and heredity. Genetics and heredity. That will be our main focus in this class. So genetics and heredity. That are generally biology. Are we together now? Which is Bio 101. Generally, when we're talking about genetics and heredity, we say that heredity is more like, the passing of traits from parents to offspring. So when we're talking about, heredity, we're generally focused on passing of traits, transfer of traits from parents to offspring. Now, when we're talking about, transfer of traits from parents to offspring, as in heredity, transfer of traits from parents to offspring. Now, what we are generally focused on is, trait is more like a character or characteristic. If you don't call it trait, you call it what? Characteristics. So, for every parent, when they produce a new individual, be it a boy or a girl, the child will have a part of the parent. Say for instance, we can get a trait of maybe color, height, behavioral changes, from either of the parents. So the transfer of those characters, when it moves from parents to the offspring, is usually called what? Usually called heredity. Are we together now? So in this scenario, what we are looking at, in terms of this heredity is this. Now, we are talking about an adult individual. Say for instance, a man, marries a woman, and then at the end of the day, they are expected to produce a child. Say man, plus a wife, right? Now, at the end of the day, they are going to produce a child, right? Which is through fertilization. Remember, in fertilization, we're talking about is the fusion of the male gamete. Male gamete here, this is male gamete, right? Male gamete plus, which is the female gamete. Female gamete. Now we're going to form a zygote. Are we together? Now, it's in this zygote that will later on develop into an embryo, later on develop into a fetus, and then later on develop into a baby. So in fertilization, we're talking about fusion of the male gamete and the female gamete. Remember that the male gamete is called the what? The spermatozoa, sperm. Are we together now? Now, and that of the female is called the ova or egg. Female is called the what? Ova or egg. So during sexual intercourse, generally, the semen will move from the man and then enter into the body of a female. Now, when the semen move from the body of a man and then enter into the body of a female, that's what we call ejaculation. That is when fertilization occurs. So there are millions of sperm that are released. Only in just one release. In one release, millions of sperm are moved from the body of a male to the body of a female. Are we together now? Now, when these millions of sperm enter into the body of a female, only one sperm out of that millions of sperm will be able to fertilize the egg that is being released, by the body of that particular female. Are we together now? Now, when that particular, sperm fertilizes that egg, that is, when we say fertilization has happened. Now, the remaining millions of sperm that has been introduced into the body of a female, the remaining will die off. Are we together now? So, when I'm talking about, heredity, we're talking about movement, transfer of trait from parents to offspring. Are we there now? Now, as we continue in this heredity, we begin to look at also what we call genetics. Now, when we're talking about genetics, we're saying that genetics is the study of heredity, genes, and the mechanism of inheritance. Genetics is the study of what? Is the study of heredity. Is the study of heredity. This is heredity. So the study of this heredity is called what? Genetics. Are we there now? That's why genetics is an important topic in biology. Why heredity is a transfer of traits from parents to offspring, transfer of characters from parents to offspring. Now, the study of this heredity and the mechanism of inheritance is called what? It's called genetics. Are we together now? Now, for us to understand genetics and heredity, there are important things we need to look at. Number one of them is called the DNA. Are we there now? Now, DNA simply means deoxyribonucleic acid. Deoxyribonucleic acid. Now, a DNA is a double-stranded structure. Are we there now? DNA is a double-stranded structure that helps us in carrying genetic information. Say for instance, every organism has a DNA. So without your DNA, you cannot carry a genetic information. So when we're talking about, remember I said genetics is the study of heredity. Are we there now? And heredity is transfer of traits from parents to offspring. And I'm saying that for us to understand genetics better, we need to understand what is meant by DNA. And I say that DNA is what? Deoxyribonucleic acid. Now, we went ahead to say that as much as DNA is deoxyribonucleic acid, this is more like a simple structure of a DNA. Let me just give you a sketch. Say we can have something like this. This can be a structure. They are usually double-stranded. Now, inside this DNA, there are majorly, three important components. Do you understand? Present in this DNA. Remember, deoxyribonucleic acid. Number one component is what we call the nucleobase. Nucleobases. Number two component is what we call the pentose sugars. Pentose sugars. Are we there now? And number three component is what we call the phosphate moiety. Please listen. This is biology, it's not chemistry. So, so listen very well. So now, these are more like the components of this particular nucleic acid. Remember that under the nucleic acid, we don't just have DNA. We have two important nucleic acids. We have what we call the DNA, and then we have the RNA. Now, the DNA is deoxyribonucleic acid, and we also have the RNA, which is what? Ribonucleic acids. Are we together now? So, deoxyribonucleic acid, ribonucleic acid, both of them are the two types of nucleic acid that we can, see in biology. But when we're talking about helping to transfer genetic information from parents to offspring, we are mainly focused on what? On the DNA. So under this nucleic acid, there are major components of this nucleic acid called nucleotides. Are we there now? And there are three components, which is nucleobases, pentose sugar, and phosphate acid moiety. Now, under these nucleobases, please listen and listen good. Under these nucleobases, we have what we call, under nucleobases are divided. Nucleobases, let's use this place. Nucleobases are divided. They are divided into three. Are we there now? Okay, let's look at the first two. Now, into two, we have what we call the purine bases, and the pyrimidine bases. The purine base and the pyrimidine base. Please listen. I've seen these questions come up in past questions. So please listen. Now, under purine base, there are three purine bases. Now, under pyrimidine, let's look at the first three in pyrimidine. Under pyrimidine, we have what we call the cytosine, we have the uracil, and we have the what? The thiamine. You can call pyrimidine CUT, cut, where C represents cytosine, U represents uracil, T represents what? Thiamine. That's nucleobase, pyrimidine base. I told you that DNA composed of three important components, which are the nucleobase, the pentose sugar, and the phosphate acid moiety. Are we together? Please, if you're following, you can let me know in the chat. Now, under pyrimidine, under nucleobase, we have two bases. We have purine base and pyrimidine base. And I say that under pyrimidine base, there are three important pyrimidine base, which are CUT, CUT. This C stands for cytosine, U stands for uracil, T stands for what? Thiamine. Now, under purine base rather, we have AG. For those of you that attend Apostolic Church, AG, or Anglican Church, however you mention it. So we say that A stands for what? A stands for well, let's look at guanine. Guanine how do you know? Now, generally, I want you to understand something. When we're talking about nucleobases, without the understanding of these nucleobases, we cannot actually form anything called a nucleic acid. Just like I've been mentioning, cytosine, which is CUT. How do you know? We cannot form anything called a particular DNA without these nucleobases. So for us to understand nucleobases, for us to understand DNA rather, we need to first of all understand these nucleobases. Remember, we're still talking about genetics. And I told you that genetics is a study of what? Heredity. And what is heredity? Heredity is the transfer of character from parents to offspring. I'm taking it one after the other. There are a lot of things that we'll be doing here. So I said that we have CUT, cytosine, and this, we have guanine, and we try as much as possible to look at the A. A is adenine. Adenine and what? And guanine. So there are bonding in each of these bases. There are questions in your, past question where they will ask you which of these bases bond together. Now we say that C, cytosine, use 3 hydrogen bond to bond with, guanine, and adenine use 2 hydrogen bonds to bond with thiamine. Are we together now? Now, these are the bases that are found in nucleic acid. But there are only, out of these 5 bases, if you look at these bases, we have 1, 2, 3, 4, 5. If you look at these bases, there are 5 nucleobases present. But if I ask you how many bases are found in DNA, it is 4. How many bases are found in RNA? It is 4. Remember what I started talking about was DNA. Under DNA now, there are 4 bases, which are cytosine, guanine, adenine, and thiamine. Now, under RNA, we have cytosine, guanine, adenine, and uracil. This is RNA. This is what? DNA. I don't know if you're following. So, I said that this is purine bases, AG, pyrimidine, CUT, adenine, guanine, cytosine, uracil, thiamine. These are 5 nucleobases, 5 individual bases. Nucleobases are divided into 2, purine base and pyrimidine. Under purine base, we have adenine, guanine. Under pyrimidine, cytosine, uracil, thiamine. So these are, under DNA, there are 4 bases. Cytosine binds with guanine. Adenine binds with what? Thiamine. If you come to RNA, remember RNA is also what? It's also a nucleic acid. Cytosine binds with guanine. Adenine binds with what? Uracil. That's RNA. But we're not looking at RNA here. We're focusing more on what? On DNA. So we are done with the first, component of DNA, which is nucleobases. Now, there is another component called what? Pentose sugar. Pentose sugars. Now, pentose sugars are, monosaccharides. Pentose sugars are what? Are monosaccharide. But before we, go deeper on, this pentose sugar, I want to explain something to you. Now, when we're talking about pentose sugars are sugars that have 5 carbon atoms. We have what we call triose sugars, we have tetrose sugars, we have pentose, we have hexose. Now, when we're talking about, pentose sugars are monosaccharides that have 5 carbon atoms. Are we there now? Example, we have the ribose and deoxyribose. They are all monosaccharides. If we come to triose sugars, let's explain something. We have triose. Please, Remember, we are talking about pentose, but I want to go deeper a bit. We are learning biology, so in case you see the question, you can be able to answer. So we have what we call triose sugars. We have tetrose sugars. We have pentose. And we have what? Hexose. Are we together now? So when we're talking about triose sugars are carbohydrates that has 3 carbon atoms. Generally, the type of carbohydrates we are focused on here is monosaccharides. They are not just carbohydrates, but they are what? They are monosaccharides. So in this scenario now, we're saying triose, under triose, we have what we call aldotriose. I'm taking you somewhere. We have aldotriose and ketotriose. Under tetrosis, we also have aldotetrose and what? Ketotetrose. The same thing with, this particular pentose. We also have aldopentose and what? Ketopentose. If we also come to hexose, we also have aldohexose and ketohexose. When we're talking about aldose sugars are sugars that contain aldehyde functional group. You can write it down. Aldose sugars are sugars or carbohydrates or monosaccharides that contains what? Aldehyde functional group. What is an aldehyde functional group? Aldehyde functional group is RCHO. That is aldehyde. Are we there now? While ketone functional group is RCOR prime. So this is ketone. This is what? Aldehyde. Are we there now? So we have tetrose, we have triose, tetrose, pentose, hexose. Are we there now? But what we are looking at in this DNA is my what? Pentose sugar. I don't just want to explain pentose sugar. I just want to touch this a little bit so that we can, dive into what we're looking at. I hope that's okay. So follow suit. Now, for my triose sugars, I say we have aldotriose and ketotriose. Let me just give a structure. When we're talking about aldotriose and, ketotriose, look at what I'm looking at in this case. Say, I told you that we have what? RCHO. R here can be a methyl. Generally, when we see R in chemistry, R is usually what? Methyl groups. I wouldn't know. So I can have a sugar like this, CHO. Remember, triose is 3 carbon atom. C. This is H, this is OH, and this is CH2OH. This is a monosaccharide. The name of this monosaccharide is, D-glyceraldehyde. Can jot it down if you want. D-glyceraldehyde. So this is an example of what? Of aldotriose. Now when we move to ketotriose also have ketone functional group. And they also 3 carbon atom. If you look here, this is carbon 1, carbon 2, and carbon 3. So it's triose. So which type of triose? Aldotriose, because it has CHO at the beginning. Are we there now? So this is an example of what? Aldotriose. We also have ketotriose. It must have 3 carbon atom, but it will have CO in between. We can have something like this, CH2OH, C double bond O, and then we have CH2OH. Are we together now? So this particular sugar, the name is what? Dihydroxy acetone. Are we there now? Dihydroxy what? Acetone. The name of this particular sugar is what? Ketotriose. This is a ketotriose. Are we there now? It's a ketose because it has C double bond O in between. Remember, RCOR prime. R starts, which is CH2O. CO comes in between, and R ends. This is an example of aldotriose and what? And ketotriose. Now, let's look at this particular example too. Let's continue with aldotetrose. Now, in aldotetrose, look up now. We're going to have something like this. CHO, remember, 4 carbon atom. 1, 2, 3, and 4. CH2OH. And then I have here HOH. I have HOH. Now, this is an aldotetrose. Tetrose, 4 carbon atom. The name here is erythrose. Please, I'm trying to explain, pentose, tetrose, and all that so that you don't get confused in case you see the question. Now, we've explained erythrose. We also have CH2OH. Let's go to, this is aldotetrose. Let's go to ketotetrose. Now, we can have something like this. CH2OH, C double bond O, C. This is H, OH, CH2OH. Are we there now? Now, the name of this particular sugar is what? erythrulose. Erythrulose. This is a ketopentose. This is an example of a ketopentose. Ketotetrose rather, sorry. Ketotetrose. Why is it tetrose? Because it has 4 carbon atoms. I don't know if you are getting what I'm saying in this scenario. Okay, now if we move to aldopentose and ketopentose. In aldopentose, remember we are in pentose. Remember what we are doing here, nucleic acid, pentose sugars. Under pentose sugars, we have aldopentose and ketopentose. So we have aldose, aldopentose. Aldopentose are aldehyde sugars that have 5 carbon atoms. We have example CHO, 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. And then I have something like this. Say I have H, OH, I have H, OH, I have H, OH. Now this is an example of what? Ribose. I also have CH2OH, and then I have C double bond O, C, 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 H2OH, and then I have H, OH, H, OH. This is ribulose. Another examples of aldopentose is deoxyribose. Deoxyribose. Ribose and deoxyribose are aldopentoses. Now, when we continue with all these sugars, we also have aldohexose. Aldohexose like your glucose, you understand, your galactose, your mannose, and then we also have ketohexose. Ketohexose, ketone functional group, but 6 carbon atom. Example is fructose. Are we together now? So in this scenario, let's go back to our pentose sugars. In this scenario, the pentose sugars that we need to form this DNA, it is what? An aldopentose, which is what? Deoxyribose for DNA. Are we together now? So now the next component of this particular, nucleic acid, which is DNA, is a pentose phosphate moiety rather. Is a phosphate moiety. Now is what? A phosphate moiety. Now this phosphate moiety is usually H3PO4. So that means, let's go back. That means in every DNA, like this structure here, for us to have this DNA, we must have these 3 components, which is the nucleobases. Which nucleobases do we see in this DNA? There are 4 of them. Remember, cytosine, guanine, adenine, thymine. So how they usually bond in this nucleic acid, in this particular DNA, is something like C to G, A to T, C to G, T to A. We'll continue A, G, triple bond to C, A to T. We'll continue until we fill up these spaces. This is how a DNA looks like. So all these simple letters you are seeing here, C represents cytosine, G represents guanine, A represents adenine, T represents what? Thymine. So DNA is double-stranded, whereby RNA is just single-stranded. This is a typical structure of an RNA, something like this. This is just RNA. Is just what? Single-stranded. Okay, so I've explained DNA, and I told you that they are responsible for carrying what? The genetic information. They are responsible for carrying the what? The genetic information. Now, there are what we call genes and traits. As a biology student, we need to understand what we mean by what? Genes and traits. Now, when we're looking at, genes and traits, what we are generally focused on, we are trying as much as possible to look at, how characters are coded, and how these particular characters are being moved from parents to what? Offspring. That is what we call genetic coding. In biochemistry, for those of you that will be studying biochemistry, you begin to learn about what we mean by what? Genetic codings. How a character, how a particular genetic information is being coded, in the body of an organism. Are we together now? Now, it's in these genetic characters that you understand what we mean by, inheritance patterns. Say, for instance, an individual. Let's take, for instance, let me show you something. Let me show you something. I can get an individual, say, let's say we have a man, and then a woman. Remember, that's what genetics is all about. It's all about reproduction, the characters they transfer. You understand? So in this scenario now, we have a man and a woman. And if, let's take for instance, this is a man, and then this particular man is we are creating a scenario here, right? He's for it to be a man, he's old enough, a woman old enough too. Now I want us to look at inheritance patterns. Now under this inheritance pattern, we have what we call characters that are dominant. We have what we call dominant characters. We are coming back here. We have what we call dominant characters. Dominant characters. Are we there? We have what we call recessive characters. Recessive characters in this man and woman. Are we there? We have what we call homozygous characters. Homozygous characters. Please, if you are following, you can just indicate in the chat. Let me know that I'm talking to a group of persons. We have homozygous characters. We have heterozygous characters. We have heterozygous characters. We have what we call a genotype. A genotype. We have a phenotype. I will explain them one after the other. Just work with me. We have a phenotype. We try as much as possible to have, let's say, alleles. Let's add alleles. Let's add alleles into the play. Okay, and then maybe let me add chromosomes. Okay. Now, let me explain. And when we're talking about a dominant character, now for us to say that a character is dominant, what we are saying is this. Say, for instance, we have, let's say we have a tall character. Let me use tall and short. Tall and short. A dominant character is usually represented with capital letter. Are we there now? A dominant character is usually represented with what? A capital letter. If I have any character that is represented with capital letter, that particular character is what? Dominant. And a dominant character is also a character that expresses itself in a particular offspring. Are we there now? Despite it being a homozygous or heterozygous condition. I will explain. So that means I can have something like this, capital T, small t, and capital T, capital T. Look up now. Capital T, small t, this is the heterozygous because I have two different letters here. Capital T and small t are two different letters. Are we there now? Why capital T is dominant, small t is what? Recessive. Why capital T is dominant? Capital T here is also what? Dominant. So that means a dominant character is a character that expresses itself either in a homozygous or what? Heterozygous condition. They are usually represented with what? A capital letter. So between tall and short, which one do you think is dominant? Tall is dominant over short. I don't know if you're getting what I'm saying. Now, if you're talking about recessive characters does not show itself either in homozygous or heterozygous conditions. Recessive character only shows itself only in a homozygous condition, like small t, small t. Are we there now? So this is a recessive character, usually represented with what? Capital letter. Dominant characters are usually represented with what? A capital letter. Recessive, a small letter. I don't know if that makes sense. I said recessive characters are represented with what? Small letters. Dominant characters are represented with what? Capital letters. A dominant character shows itself or manifests itself either in a homozygous or heterozygous condition. But in a recessive character only shows itself in what? A homozygous condition. When we're talking about homozygous character, we're talking about homozygous condition and heterozygous condition. So let's look at homozygous characters. Now, when we're talking about homozygous characters are characters that contains pair of similar alleles. Pair of similar alleles like capital T, or small t, small t. Pairs of similar allele, similar, capital T, they are the same. Small t, they are the same. That is what? A homozygous character. When we're talking about heterozygous characters are pair of the similar allele, capital T, capital A, small a. Capital letter and small letter, whenever you see them together, they are what? Heterozygous characters. I said in dominant are represented with capital letter. They are present in a particular generation or in a particular child, either in a homozygous or what? Heterozygous condition. Meanwhile, in recessive characters, they are present in a child or in an offspring only in a what? A homozygous condition. Now, what is a homozygous character? A homozygous character are pair of similar alleles, pair of similar, either capital T, or small t, either capital A, or small a, either capital Y, or small y, either capital G, or small g, either capital B, or small b. They are what? Homozygous character. Homo, the same, pair of similar alleles. Now, when we're talking about heterozygous characters are pairs of dissimilar capital T, small t, capital A, capital B, small b, capital Y, small y, capital G, small g. I don't know if you get what I'm saying. So I've explained dominant, recessive, homozygous, and what? Heterozygous. So when I say a particular character is dominant, but in homozygous condition, when I say dominant, you should know that we are talking about capital letter already. Dominant is capital, but I say dominant homozygous character, it's capital T. If I say dominant heterozygous character, it's capital T, small t. If I say recessive homozygous, recessive, small letter, homozygous, I don't know if you are following what we are doing here. So that is that. So now let's move on to genotype. Now, when we're talking about genotypes, we say that genotypes are genetic constitution of an organism. Genotypes are what? Genetic constitution of an organism. Now, genotypes are not what you see. They are what makes up an organism. Are we there now? Genotype are what makes up an organism. Like an individual's blood group, that's a genotype. Are we there now? The rhesus factors, those are genotypes. You don't see them with your crocodile eyes. You don't see them with your naked eyes, do you? No, you don't. So, but what do? You only see them make manifest in the body of an organism. Are you getting what I'm saying? So, genotype is more like the genetic constitution, what makes up an organism. How do you know? Genotype are what makes up what? An organism. How do you know? So, genotype can be capital T, small t. This is a heterozygous store. This is heterozygous store. Heterozygous, dissimilar allele, pairs of dissimilar, capital T, small t, dissimilar, heterozygous store. So this is a genotype. You don't know what capital T, small t means. But in phenotype, you just say this is store, a tall individual. What are you seeing? Is store. Meanwhile, in genotype, we have small t, small t. A homozygous recessive character, which is short. Small t is short. But what are you seeing? You are seeing short. So, why genotype is the genetic constitution of an organism? A phenotype is the outward expression of that genetic condition or that genetic constitution. So, you can't see genotype, but you can see what? Phenotype. Phenotypes are what you see with your naked eyes. I don't know if you are making sense here. So, what you can see an individual's ability to roll tongue. You can see the color of the skin of an individual. You can see the height of an individual. Those are phenotypes. Genotypes are what makes up those things that you are seeing. So for an individual to be fair, what causes it is genotype. The constitution of that. Are we together now? Now when we're talking about alleles are pairs of contrasting characters. Are we there now? That's alleles generally. So an allele can be capital A, small a. Those are alleles. Capital T, small t, capital T, those are alleles. Are we together now? Why this is capital letter is dominant, small letter recessive. Why this is a homozygous, this is heterozygous, homozygous. Remember, homozygous, pair of similar allele, heterozygous, pair of dissimilar alleles. So this is an allele, 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 this is an allele. This is what? An allele. Now, when we're talking about chromosomes, very, very sensitive. generally, I want you to, try as much as possible to understand something. Chromosomes are 46 in number in the body of an organism. In every male and female, there are 46 chromosomes. As you are a guy watching this video, you have 46 chromosomes. Tomorrow, when you marry, your wife will have 46, or she already has 46 chromosomes. So, generally, we have 46 total chromosomes in the body of every living organism. Now, out of those 46 chromosomes, we have 23 pairs. We have 23 pairs. For us to have pairs simply means like your shoe. You say pair of shoe, your left and the right. A single pair is 1 and 2. Now, so when we have 23 pairs, so that means 23 times 2, which is what? 46 chromosomes. So every human has what we call what? 46 chromosomes. Now, inside these 46 chromosomes, there's what we call sex chromosomes. That's what we call what? Sex chromosomes. Now, under sex chromosome, we have what we call male sex chromosomes, and the female sex chromosomes. Now, for the male sex chromosomes, we call it the XY. For the female, X and X. Remember, chromosomes are usually in pairs. So this is a pair of a male chromosome. This is a pair of what? A female chromosome. Since we have 23 pairs, this is just a single pair. If we bring out a single pair out of 23 pairs, we still have 22 pairs remaining somewhere else. So this is just a sex chromosome. We have other chromosomes. So the total chromosomes in the body of an organism is 46. How many pairs? 23 pairs. Not 2 pairs, 23 pairs. Don't forget. Are we together now? 46 and 23 pairs. And I hope you are writing these things down. You're not just watching. Please be using your notes. Now, in this scenario, I want you to look at something. If we have XY as a male sex chromosome, and then we have XX as a female sex chromosome, whenever they have an intercourse, say husband and wife, they have an intercourse, maybe this night. And then remember that there is usually a probability that this man will either give the X chromosome or the Y chromosome. And also there's a probability that this lady will give the X or the X. It's just like saying it's either I fail or I fail. That means the only option you have is failing. Or you say it's either I win or I win. The only option you have is winning. That's what we are seeing in this female. Female will always give X. So it's either they give X or they give X. I don't know if you are getting what I'm saying. So in this scenario now, what we are trying to focus on is this. So I'm saying that whenever we have an intercourse, be it a man and a woman, a child will be produced, a child will be formed. The child that will be formed will be either male or a female. So when the man brings the X and the lady brings the S, the child that will be formed will be XX, which is female. A daughter. You've produced a female child. Wow, congratulations. What's the name of your daughter? Her name will be Lucifer or Lucy. Her name will be Jennifer or Jenny. Her name will be Jezebel or whatever. Now, when the man gives the Y chromosome and the lady gives the X, we're going to form XY, which is the what? The male. So, in most jam question, they can ask you, sex determination is, dependent on the father or the mother. It's dependent on the father. Because whenever an intercourse happens, the lady will generally give out the X. It is the male that will either give the X or the Y. So when the male brings the X, so we have XX, will form a female. When the man brings the Y, we have XY from the male. I don't know if you're getting what I'm saying. So sex determination is not, dependent on the woman, but rather the male. So if in your village, when you get married to a lady and then your village people is telling you, divorce this lady, she has not produced a male child up till now. She's not fertile enough. Just know that as a man, you are the issue, you are the problem. So far as she's getting pregnant and she's producing children, for her not to give a male child is not really her fault. It's the man's fault. Because it's what you give to her that she will give back. It's what you give to her that she will produce. Are we making sense now? Good. So that is that about some of the terms associated here in this inheritance of acquired characteristics. Now I want us to also look at something else. I want us to try as much as possible to look at some of the traits and how these traits are transferred. Remember that, uh, let's take for instance, this is a man, this is a woman. I was saying something initially, and then I say when they get married, they are expected to produce a child. How would you know? Say for instance, the man is tall, the lady is short. When they marry, they are going to produce children. Some will be tall, some will be short. How would you know? But in most cases, they can produce a particular organism that is not even in their, plan, or that is not even in their, ancestral trees. That's what we call ancestral trees. Ancestral trees. If you don't call it ancestral trees, you call it pedigree trees. Pedigree. Pedigree. Are we there? Now, pedigree simply means, say for instance, there are two ladies. We have Annabelle and Jessica. Annabelle and Jessica. Now, Annabelle can be from, a particular state in Nigeria. Jessica, another state in Nigeria. Maybe Annabelle is from the west, Jessica is from the east. Now, if Annabelle is fair in complexion and Jessica is dark, say Annabelle is fair. Please look at this scenario. And Jessica is dark. Now, when Annabelle marries tomorrow, maybe she marries let's say a fair man, and then they produce an entirely dark child. The first thing we look at is what we call the ancestral tree or the pedigree tree. Ancestral tree is like generational tree. Say, okay, Annabelle, since you are fair and your husband is very fair, how come you are producing a dark child? What is the result? When we look at the ancestral trees, we look at the generations of Annabelle and the generations of the husband, and we are not seeing any dark individual. Are we there now? That might be the reason why the child or all their childs are very dark. We can call those phenomena as what? Mutation. We can call that phenomenon what? Mutation. Now the question is, what is the mutation? Now we say that mutation is the sudden change in the DNA sequence of an organism. I've explained DNA here. So when you hear about mutation, we say that mutation is the what? The sudden change of a DNA sequence of an organism. Say for instance, a lady is pregnant. Instead of her to give birth to a child, she's giving birth to an albino. A child that should be fair or dark, but she gives birth to an albino. Albino is a result of mutation. So, or she give birth to maybe an imbecile. Those are mutation. Or she give birth to an individual that's supposed to have 5 fingers, and then the individual has 6 fingers. Those are mutation or genetic disorders. If you don't call them mutation, you call them genetic disorders. I don't know if you're getting what I'm saying. Now, in this scenario, mutation can be caused, maybe it can be as a result of natural cause or even artificial cause. Artificial cause can be through, my environment, what I consume, the foods that I eat, the types of, alcohols that I drink. Most of pregnant women, when they're pregnant, they will go to hospitals and then they will subject themselves to UV radiations, X-rays and all. Those particular substances can cause mutation. It will change the way the baby is to be formed in the womb. So instead of forming a normal individual, you produce a mutated child. So there will be sudden change in the DNA sequence of that particular gene. So instead of you to produce a child, that have the characteristics of the dad, you are going to produce a new organism entirely that have a different characteristics. That's mutation. Are we together now? So in a nutshell, what I'm trying to say is, as much as we are talking about inherited characteristics, characters moving from parents to offspring, we also need to understand there are scenarios, there are conditions whereby an individual might not really have, the characters of either parent, nor, the father nor mother. That is when we begin to look at what? A mutation. Are we together now? Now, according to Mendel introduced a particular cross called monohybrid and dihybrid cross. In biology, Mendel introduced a cross called what? Monohybrid and what? And dihybrid crossing. Monohybrid and what? And dihybrid crossing. Monohybrid and dihybrid. And dihybrid cross. Alright, please, if you have any question, you can drop in the comments. Let me know if you are following me. And if you can hear me too, please let me know so that I know I'm not talking to myself alone. Are we there now? Mm-hmm. All right. Okay. If we look at monohybrid cross, we see that monohybrid cross is actually a cross. We used to study the inheritance of a single pair of alleles, a single pair of character. Monohybrid, mono, one. We used to study a single pair of character. A single pair of character can be, a height of an individual, height of an individual. Are we together now? Color of an individual, color of an individual. Are we there now? That's a monohybrid cross. We must look at what is a cross that involves the study of a single pair of character. Are we there now? A single pair of allele. Meanwhile, in dihybrid, we are looking at a cross that involves two pairs or two different forms of characters. Are we there now? Or alleles. Like I can say height and color. That is a dihybrid. I can say height and shape. That's dihybrid. Are we together now? I can say, maybe seed color and flower shape. That's dihybrid. But the monohybrid, I will just look at the height or I will look at the color. Are we there now? Say for instance, in monohybrid, they can say a man which is homozygous tall, please follow me, homozygous tall, marries a heterozygous, okay, no, let's use marries a homozygous short woman. It is love, right? Good. Marries a homozygous short woman. What is the genotypic and phenotypic ratio of the above cross? Now, first of all, we need to understand what is homozygous tall. Homozygous tall, I told you that when we're talking about tall is a dominant character, so it's capital T. Are we there now? So if it's homozygous, capital T, times. What is a homozygous short? Homozygous short, small t. So it's a recessive character, small t. Are we together now? Now, first of all, they say that we should identify the genotypic and phenotypic ratio. So what we are going to do is that we're going to use this capital T. We cross with this. I'm going to have capital T, small t. I use this particular T again to cross with this particular small t, capital T, small t. I'll now move on to the second capital T. To here, capital T, small t. Here, capital T, small t. Now, this particular generation is called F1 generation. If you don't call it F1 generation, you call it what? First filial generation, according to what? According to Grigor Mendel. It is called the what? F1 generation. So, we cannot get our phenotypic or genotypic ratio. Remember, genotype are the genetic constitution of an organism. Phenotype are the physical expression, outward expression, what you can see. Are we there now? Good. So for us to get our phenotypic and genotypic ratio, we'll have to cross to get our F2 generation. And to get your F2 generation, you self-fertilize. You take the first offspring and the last offspring. That's when we have capital T, small t, times capital T, small t. Are we together? I want to calculate our genotypic and phenotypic ratio. So what I'll do is that I will now cross. I will say capital T, we have capital T, capital T, small t, capital T, small t, small t. Now look up. If you look at this crossing, I had capital T times this capital T times small t, capital T, small t. Now we have small t times capital T. In any character, you can never ever see, a dominant character come last before, a recessive. Dominant always comes first before the what? Before the recessive. Dominant always comes first before the what? Before the recessive. except in a separate occasion, which I will show you something, just follow suit. Just be attentive. So this is what we call the F2 generation. And it's in F2 generation that we'll find our, genotypic and phenotypic ratio. Now, the phenotypic ratio is what we can see. What can we see? This is tall, this is tall, this is tall. Why this is homozygous tall? We have homozygous tall. These two are heterozygous tall because they have small t in between. Heterozygous character, pair of the similar allele. So this is homozygous tall, this is heterozygous tall. So the ratio is 3:1. Are we there now? Because we have 3 tall and 1 short. 3 tall and what? And 1 short. So the phenotypic ratio is what? 3:1. And the genotypic ratio is now, what we are seeing here is capital T, capital T. How many capital T, capital T do we have? We just have 1. So it's 1:2. And then we have capital T, small t, capital T, small t into 2. And then we have small t, 1. So the phenotypic ratio is 3:1. And the genotypic ratio is 1:2:1. I don't know if you are following what I'm doing here. So now, in this scenario, this is an example of a monohybrid cross, because in monohybrid cross, we are only focused on one separate character. We are only focused on what? On one separate character. As in dihybrid is 2. We are coming to dihybrid. Let's finish up with monohybrid. Are we there now? So we talked about height. Let's look at, another, monohybrid. Let's say something like a skin color. Now, in most time, whenever we come down to this particular F2 generation, it is only when they ask us for, genotypic and phenotypic ratio, or the genotypes and phenotypes. But if they just ask you, say, cross between, say, for instance, they say, what are the, products of the children, or what are the number of children that are formed when, say, an individual that is heterozygous tall, let me use an individual that has heterozygous, trait for a red skin. Let's say heterozygous red skin. No, do we have red skin? Let's say fair skin. Let's use fair. Please, all these things are coming from my head, so don't hear me. Let's say heterozygous fair skin, F-A-I-R, heterozygous fair skin, and let's say homozygous dark skin. Are we there now? And then they ask me, find the result. And they give in brackets, note that fair is dominant. Note that fair skin is what? Is dominant. And when they say heterozygous simply means heterozygous dissimilar. So if fair is dominant, that means I'm going to have, let me say fair skin. Let me use fair skin as, let me use it as, capital B. And then use dark skin, which is heterozygous, because if fair is dominant, dark will be recessive. And then let me say dark skin as small b. Are we together now? Now, in this scenario, let's take, for instance, we're going to do something like this. So in my fair and dark, I'm going to have, because they say heterozygous fair skin, remember, heterozygous fair skin, I'm going to have capital B, small b. That's heterozygous fair skin. Heterozygous fair skin. And then they said, cross with homozygous dark skin. Homozygous will be capital small b, homozygous dark skin. So when they cross, I'm going to have capital B, small b times small b. Now in this scenario, they didn't ask us for genotypic or phenotypic ratio. What they just ask us is, what is the result of the offspring? So what I'm going to have here, I'm going to have capital B, small b, capital B, small b, small b, small b, small b, small b. Are we there now? So I have 50% of what? 50% of, fair skin, of heterozygous fair skin, and 50% of homozygous what? Dark skin. Now, if I don't want to use this cross, I can simply use a Punnett square. There is a square called what? Punnett square. Punnett square. This is the one we usually use in solving for our dihybrid cross. So my Punnett square, what I can easily do in Punnett square is that I will just form a block, something like this. I'll form something like this as a block, and then I have this, then I have this. I will come here, I will take each of these genes, I will say capital B, small b, and then I'll bring capital small b, small b. So I will say capital B times small b, I will have capital B, small b, small b, small b, small b, small b, capital B here, capital B, small b, small b, small b. So that means I have 50% this, 50% this, which is the same thing as this. Are we there now? So this is a simple Punnett square. If I don't want to be crossing it this way, I can use this Punnett square to simply solve and still get my answer. Are we there now? So the offspring that was formed are 50% of heterozygous fair skin and 50% of what? Of homozygous dark skin. So these are questions on monohybrid crossing. In monohybrid cross, what we just have to do is just, we'll cross and then we identify our phenotypic and genotypic ratio. Generally, in monohybrid cross, the phenotypic ratios are usually 3:1. Are we there now? And the genotypic ratio are usually what? 1:2:1. I don't know if you're getting what I'm saying. So that is that on monohybrid. I want us to look at dihybrid cross. I hope we're following. All right. Now for dihybrid cross is a bit complex a bit. So but just follow suit to understand. And in most cases where you begin to look at your JAMB questions, there are a lot of questions in JAMB, where we begin to look at all this monohybrid and dihybrid. There are a whole lot of them. Are we together now? So you can ask in the comment section, or you can ask in my DM. But let's come here now. We are done with monohybrid. Let's go to dihybrid. Dihybrid. Dihybrid cross. Let's use a seed. We remember we talked about two characters, right? Seed color and seed shape. Let's use seed color and seed shape. Are we there now? Let's take, for instance, in the seed color, we can have, wrinkled and green. So we can have, let's say color, because we're not talking about shape. Color, we can say green and yellow. Green and yellow. That's for color. And then for shape, we can have round and wrinkled. Are we there now? Let's say the green is dominant, yellow is recessive, because in every particular character, we must have a dominant and a recessive. Like when we have tall and short, tall is dominant, short is recessive. Are we together now? In this scenario, when we're talking about, what's it called? We are generally talking about, green and yellow, round and wrinkled. We're talking about, color and the shape. Are we together now? So in this scenario, what we're going to do here is this. We're going to try as much as possible to identify the one that is dominant. So green is dominant. So if green is dominant, I'm going to give green as capital G. If yellow is dominant, I'm going to give yellow as what? Small g, recessive rather. Green is dominant, yellow is what? Recessive. So this one is going to capital G, and this one is going to be what? Small g. And if I come to round, I'm going to give round as capital R, and wrinkled is going to be recessive. Are we there now? Because round is already dominant, so wrinkled is recessive. So I'm going to give wrinkled as what? Small r. Are we there now? Now, generally, what happens is this. We will try as much as possible to get out the characters. Are we there now? Let's look at, this is color. Are we there now? Green and yellow is the color. Are we there? Green and yellow is color. And then round and wrinkle is shape. Are we together now? So if we come here, G and G, what we're going to have is capital G, small g, small g. And if we come here, we're going to have capital R, small r, small r. So the first step is that we are crossing, this is the hybrid crossing. They can ask us to identify or show the phenotypic and genotypic ratios. But generally, they don't usually ask genotypic in dihybrid. They usually focus on what? On phenotypic ratios. So what we're going to do here is that, well, first of all, we're going to identify, say we have something like capital G, small g, times. This is already a specific character. Which character is this? Color. Times the what? Times the shape. What is the shape? Capital R, small r. How do you know? So in a nutshell, what I'm going to do here is this. I'm going to take out one of this particular character, one of this particular character, one of this particular character, one of this particular character. I don't know if you are getting what I'm saying. I hope I'm not confusing you, Abi. Just go through it again. let me give you some seconds to assimilate what I've been saying. If you have any question, please just let me know. Oh. Are we there now? So we've identified the color and the shape. Now, the next step of what we're going to do here is this. Now, before we even start separating the characters, we'll try as much as possible to take dominant separate, recessive separate. All dominant will come together and all recessive will come together. So what are the dominant? Capital G, capital R, and small g, small r. Let's turn over to follow up. Please don't be distracted. Say I have capital G, how do you know? Capital R, times small g, small r. I've, separated the dominant. I told you that all dominant will come one side, all recessive will come the other side. Because already we have here now, we have green and round. We have, which color is this? Let's check back the color. Yellow and wrinkled. These are recessive characters. These are dominant what? These are dominant characters. Now the question is, what is the next thing that I'm going to do from here? Now, in my next step that I'm going to do here is that I'm going to try as much as possible to take one here, take one here, take one here, take one here. Like, this is the result of my offspring. I'm going to have something like this. Remember, we're still talking about, dihybrid cross. So I'm going to have something like this. Capital G, small g. Are we there now? Capital R, small r. 1G combined with small g. 1R combined with what? Small r. Now, what I'm going to do is I'm going to use this now to form, the 4 offspring that I'm going to use in my gametic Punnett square. So I'm going to use this particular man, capital G, small g, times capital R, small r. I will cross them together. So when I cross, I'm going to have something like this. Capital G, capital R, capital G, small r, small g, capital R, and then small g, small r. I will use only these 4 offspring, and then add it both sides on my Punnett square to actually get my phenotypic ratio of my dihybrid cross. Are we there now? So what I'm going to do here is this. Please look up. Don't be distracted. What I'm going to do is I'm going to have something like this. GR, small g, capital R, small g, small r. I will come here. Capital G, capital R, capital G, small r, small g, capital R, and then small g, small r. I will create, I told you, we're talking about what? A Punnett square. So please look up. This is how we do a Punnett square. All these things, this garment here, I will write it on a straight line and also write it on this line. So I will come here, then I will arrange, and arrange. So I will start, crossing. This times this will multiply with this will multiply with this will multiply with this will multiply with this. So if I come here, I'm going to have capital G, capital R. If I come here, I'm going to have capital G, capital R, small r. Are we there now? Capital G, capital R. That's what we have here. Here, capital G, capital R, small r. Remember, I told you that, dominant will come first before recessive. So here now, I will come here also. Capital G, small g, capital R, capital R. I'll come here, capital G, small g, capital R, small r. Are we there now? Now we'll go to the next line. Capital G, capital R, small r. Are we there now? We'll come here again. Capital G, small r. We come here, capital G, small g, capital R, small r. We come here, capital G, small r. We come here, capital G, small g, capital R. Here, capital G, small g, capital R, small r. We come here, small g, capital R. We come here, small g, capital R, small r. We come here, which is the last part here, capital G, small g, capital R, small r. We come here, capital G, small r, small r. We come here, small g, capital R, small r. I hope I'm not making mistake. We come here, small g, small r, small r. Now, it is in this Punnett. This particular square is called a Punnett square. Punnett square. Are we there now? It's in this Punnett square that will help us identify, the phenotypic ratio of this particular man. So let's look at the characters that were formed. We have capital G and capital R. Are we there now? Capital G and capital R. Remember, capital G and capital R simply means either capital G, small g, or capital R, small r, or capital G, or capital R, small r. We are still the same thing. So for us, it starts with a dominant and a dominant character, a dominant color, a dominant shape. So that is, what is a dominant color called? Let's go back to our question. A dominant color G is green. So green, and capital R is what? Is a dominant shape. R is round. So green and round, we have green and round here. We also have capital G and small r. So we also have capital G and small r. Capital G is green, small r is wrinkled. I'm trying to identify the separate characters that were formed here. So green and wrinkled. Let's go. Let's start from the beginning. I say we have capital G, small capital R. Here is capital G, R, G and R. Are we there now? Capital G and R, capital G and R, capital G and R, until we come here, capital G and small r, which is what I just wrote. Are we there now? Capital G, capital R, we already have it here. Capital G, small r, we already have it here. Capital G, capital R, we already have it here. Capital G, capital R, we already have it here. So we have small g, small r, small g, capital R, small g, capital R. That is wrinkled, and wrinkled. No, small g is not wrinkled now. Small g is yellow. Thank you. Small g is yellow. Color is yellow. Yellow and round. Yellow and round. So we have small g, capital R, small g, capital R. We have small g, small r. So small g and small r will be yellow and what? And wrinkled. Are we there now? So let's count how many green and yellow that was formed here. How many green and yellow was formed in this scenario? So for us to count how many green and yellow that was formed here, what we're going to do is that we'll say here, this is 1, capital G, capital R, 1, capital G, capital R, 2. Let me start counting it. 1, 2, capital G, capital R, 3. Are we there now? What we are mainly focused on is dominant. Are we there now? The first letters that start with each character. So capital G is dominant. It must overpower recessive. I've told you severally. If I have a dominant character and a recessive, I will only choose what? A capital letter over the small letter. The recessive character only manifests itself only in a recessive condition, only in a homozygous condition. Recessive character manifests itself only in what? Only in homozygous conditions. So here now, here we have G and R, 1, G and R, 2, capital G, capital R, 3, capital G, capital R, 4. We go to the next one, capital G, capital R, 5, capital G, small r, no, let's leave out, capital G, capital R, good, 6. We're only looking for capital G, capital R, 6. Here, capital G, capital R, 7. Capital G, capital R, 8. No. Okay, 9. Are we there now? Is there any other capital G, capital R? No. So that means green and round is 9. If we look at green and red code, which is capital G, small r, we start from here, which is A. Let's use A. Capital G, small r. Let's use B. That's number 2. We'll come here again. Check. Help me check. Capital G, small r. C, which is 3. Abi? So we have 3. Are we together now? So if we come here to yellow and round, yellow should be small letter G. So we start looking for small letter G. We have A, we have B, and we have C. Look at it. So it's also 3. And yellow and red code, which is small g, small r, 1. So that means the phenotypic ratio here is 9:3:3:1. That's the phenotypic ratio of a dihybrid cross. The phenotypic ratio of a monohybrid cross is 3:1. The phenotypic ratio of a dihybrid cross is 9:3:3:1. That means the total, characters or the total, offspring form are what? 16. Are we there now? So this is a typical example of what? Of a dihybrid cross. Should we do one more example? Now, for us to actually look at, let's try one more dihybrid cross. Let's try one more dihybrid cross. Let's take, for instance, if we have, they can give you, say, let's use height and color. Let's do this particular dihybrid for, let's use our understanding of, okay, let's use a plant. Let's use a flower color, and a flower height. So let's use height and color. Height and color. Height and color. The first thing we'll do is that we need to understand, when we're talking about height, what are we looking at? Height is usually tall and what? And short. The color that we're looking at is usually, let's look at the colors. Now, the colors that we have generally focused on, sorry about that. Okay, I believe you can see what I'm writing here. Now, the colors are usually, let's take, for instance, let's take black and white. Let's take black and white. Are we there now? Let's take black and white. And remember, black will be dominant. Tall will also be what? Will be dominant. So that means for our height, this is another dihybrid example. This is example 2. Example 2. Are we there now? We want to get the dihybrid ratio. Let's prove here. So the next thing you will do is that you go to your height. You identify, say, height, we have capital T, and what? And small t, small t. This is tall, this is short. And then for color we'll have capital B, and what? Small b, small b. Are we there now? So that means capital B is black, small b is what? Is white. So that means capital B is dominant, small b is what? Is recessive. I don't know if you are getting what we are doing here. So in this scenario, what we're going to do is that we'll try as much as possible to take, the dominant character separate, just as I told you before. So for us to take the dominant character separate, what we're going to do is that we're going to take capital T, capital B, times small t, small b. So dominant separate, recessive separate. Now when we are done here, I will try as much as possible to pick out one dominant, are we there now? And then bring out one recessive that looks alike. One dominant, one recessive that looks alike. Say, for instance, I'm going to have the result here. Capital T, small t, capital B, small b. Are we there now? So I can easily cross to get the offspring that I will use to form my what? My Punnett square. So I'm going to do something like this. Capital T, small t, times capital B, small b. Why B is black, small b is white. Why capital T is tall, small t is what? Is white. So it's rather, why capital T is tall, small t is short rather. So what I'm going to have here, I'm going to have capital T, capital B, capital T, small b, small t, capital B, small t, small b. Are we there now? So I will use this to form a, a Punnett square, just as I told you. Are we there now? So what I'm going to do here is this. Let's turn over the other side. Can we use this? Okay, let's turn over because of our stuff. So what are the characters I have here? I have capital T, capital B. Let's write it down. Capital T, capital B. Which other one do I have here? I have capital T, small b. Capital T, small b. Which other one do I have here? Small t, capital B. Small t, capital B. And small t, small b. Are we together? Can we see it here? Okay. Now I'll also repeat the same thing here. Capital T, capital B. Abi? And then capital T, small b. Small t, capital B. Small t, small b. And then I will cross. I will make a space. Are we there now? And then this is what I'm going to have here. Something like this. Something like this. And then I will start crossing. This times this. This is more like multiplication, if you can, do it so that you don't forget. So you cross, we're going to have capital T, capital B, capital B. Are we there now? Capital T, capital T, capital B, capital B. Cross here, capital T, capital B, small b. We cross here, capital T, small t, capital B, capital B. Cross here, capital T, small t, capital B, small b. Are we there now? You cross here now. Capital T, capital B, small b. Here, capital T, small b, small b. Here, capital T, small t, capital B, small b. Here, capital T, small t, small b, small b. Here, capital T, small t, capital B, capital B. Here, capital T, small t, capital B, small b. Here, small t, capital B, capital B. Here, we have small t, capital B, small b. Here, we have capital T, small t, capital B, small b. Here, capital T, small b, small b. Here, small t, capital B, small b. Here, small t, small b, small b. So we have to identify the different, characters. Remember, dihybrid, two separate characters. Dihybrid, two separate characters. That's why we're having, two characters here. Had it been is monohybrid, it would just be one character, simple character. Either it's capital T, small t, or capital B, small b. But here we have what? Two separate characters. So let's identify the phenotypic ratio. So we have capital T and capital B. Capital T and capital B, what is it known as? Capital T, remember we said that capital T is what? Is tall. So tall, and capital B is black, because B is what? Is dominant. So we have capital T, this is capital T, capital B, capital T, capital B, capital T, capital B, capital T, capital B, capital T, capital B. Okay, we move here, capital T, small b, capital T, small b. What is capital T? Tall. What is small b? Small b is what? Is white. Are we following? So small b is what? Is white. So we move here, small t. Okay, no, before we move to small t. So we have capital T, small b, capital T, capital B is already here, Okay, small t, capital B. What is small t? Small t is short. What is capital B? Black. Are we there now? Because black is dominant. Now we have small t, small b. Small t, small b. What is small t? Short. What is small b? And white. Abi? So let's count the number of tall and black, which is capital D and capital B. So we have here 1, 2, 3, because you can see capital T, small t, capital B, small b. I told you that we usually focus on the first letters. I would then the first letters in each character that will help us to understand, the particular, characters that we have. So this is 4. Capital T, capital B, small b, 5. Capital T, small b, no. Capital T, 6. No. 7. Because capital T, small t, and capital B is still capital T and capital B. The first and the first letter of the first character, the first letter of the second character. Here is capital T, capital B, which is 8. No. Now here, which is 9. So we have tall and black is 9. So let's look at capital T and small b. The first letter must be capital D. The second character, the first letter also will be what? Small b. So we come here. This is capital T, small b, small b. So this is the first letter here is capital T. And the second character here is small b. So this is 1. Let's say A. Capital T, small b. Look up here. This is also B. Let's look for capital T, small b. This is C. Is there another one here? So it's 3. And then we come for short and black. Short and black will be small t, capital B. So we start looking for small t. Look at small t. Let's say I. Are we there now? Small t, capital B. And triple I is also what? 3. And small t, small b is just this particular last one, which is what? Which is 1. So the phenotypic ratio of this dihybrid cross is 9:3:1. Are we together now? So that is the phenotypic, ratio of this particular dihybrid cross. So what you can do is solve other questions relating to dihybrid cross and monohybrid cross. 9:3:1, which will give us what? 16, results that we have here. So I think that is all about this particular, genetics. Maybe in our next biology class, we'll be looking at, evolution. Look at evolution, look at variations, and then we'll look at what? Adaptation. So if you have any question concerning each of these particular topics I've, been able to, explain here, talk about, the concept that I've just, given out, try as much as possible to, drop it in the comment section. And then if you check the past question and then you see similar questions, but you can't be able to solve, also let me know. Are we together now? I can see, ways that can help provide directions to, make this particular course to be, better. Now, also, I've mentioned what we call genetic disorders, which can, be as a result of mutations. Some of the examples of genetic disorders, we have what we call your sickle cell anemia. We have what we call your cystic fibrosis. We have Huntington diseases. They are all what? They are all genetic disorders. They can arise as a result of, the faultiness of the genes. Faultiness of a gene can also be as a result of what? Of mutation. So as a young individual, be it a young man or a young lady, try as much as possible to, practice a healthy lifestyle. Practice a healthy lifestyle so that tomorrow you will not be the issue, that we'll have. So that tomorrow also you'll be able to help produce a stronger individual. Generally, when I was talking about genotypes, I did mention something. We have what we call ABO blood group. ABO blood group. Are we there now? These are ABO blood group. We have blood group A. Blood group A. I hope you can see what I'm doing. We have blood group A. Let me just add this to the explanation. We have blood group A. We have blood group B. We have AB, and we have O. Now, I'm not talking about their rhesus factors. Rhesus factors can be positive or negative. That's not where I'm going. But if you look at these particular blood groups, ABO blood group, this is blood group A, blood group B, blood group AB, and blood group O. Now, each of these blood groups have what we call antigen and antibody. Antigen and what? An antibody. Antigen and antibody. Are we there now? Now, when we're talking about antigen is more like a foreign substance that the body does not recognize. Are we there now? That is antigen. Now, antibody is more like, a fighting mechanism, a fighting organelles, that helps the body, to fight against this particular what? Antigen. So each blood group A, blood group B has antigens. But when we come to blood group AB and blood group O, they are, more like, rare blood groups. Blood group AB is also called what? The universal recipient. The universal what? Recipient. Blood group O is called the what? The universal donor. The universal donor. Let me show you something. When we say blood group AB is universal recipient, that means it can receive blood from blood group A, can receive blood from blood group B, can receive blood from blood group what? O. And we say that blood group O is a universal donor. It can give blood to blood group A, it can give blood to blood group B, it can also give blood to blood group what? AB. Now, let me show you something. Now, for this particular man to receive blood from universal recipient is receiving. How do you know? For this particular man to be a universal recipient, it has no antibody. Blood group AB has no what? No antibody. That is why it is what? A universal recipient. So if someone asks you, why is blood group AB a universal recipient? It has no antibody. How would you know? Remember, antibody are like fighting agents. They are like shield. You know, when you are fighting, like, you know, when you are watching all this war movie and then people are fighting, kind, shine, shine, shine. Now, when they are fighting, you know, we usually use shield to defend our body so that the knife will not cut any part of our body. So, you know, those shields are more like antibody that helps us to defend the fightings. Are we together now? So any particular, you know, individual that does not have an antibody, you begin to see that that individual will be infected easily because there will be no, you know, fighting mechanisms. Are we there now? Now, if you don't call it antibody, you call it immunoglobulins. But let's not go there first. Let's focus here. Now, so blood group AB has no antibody. It has nothing that fights against this particular blood group. So that means whenever blood group A enters an individual blood group AB, just enter PAM because there is nobody fighting. This B enter PAM because there is nobody fighting. O enters PAM, there is nobody fighting. So it doesn't have an antibody. So blood group AB has no antibody. That's why it is what? A universal recipient. Meanwhile, blood group O is a universal donor because it has no what? No antigen. It has no what? No antigen. So someone will ask you, say, why is blood group O a universal donor? It's a universal donor because it has no antigen. So when it enters into the body of these particular organisms, this blood group A is not sensing any chemical substance that is not recognized. So it's more like a new blood. The same blood is entering. Because it has no antigen, it will be easier to move to blood group A, enter blood group B, enter into what? Blood group O, AB rather. Are we together? So blood group AB is a universal recipient because it has no what? No antibody that help to fight against the activity of this other blood group. And blood group O is a universal donor because it has no what? It has no antigen. And I've told you what are antigens. Antigens are chemical substances that are not recognized by the body. Are we there now? Most time the body produces an antibody against it. What is an antibody? An antibody is like a fighting mechanism. Are we there now? It's like a shield, a fighting mechanism that keeps, you know, antigen away from the body. So this is blood group AB and this is what? Blood group O. So I believe we've actually done quite, you know, few things here in biology. So in my next biology class, just as I said, we'll be looking at variation, adaptations, and what? And evolution. So I would like to see you in that class. Don't miss, and have a nice day. Thank you for your time.